The layer architecture of the neocortex is mainly composed of PC-type cells. The complex network of synaptic connections that links them can undergo persistent changes as a function of external stimuli and internal dynamics. This process is thought to underlie learning in the brain, and it is known as synaptic plasticity. Physically, plasticity can manifest as LTP or LTD of synaptic efficacy, which are thought to be mainly driven by postsynaptic calcium transients. Studying the work of Kindem et al., we developed a simplified plasticity model, implementing a neuron model to simulate pre- and postsynaptic neurons, and a synapse model to simulate a single synapse. The Hilton-Oni model was considered as a starting point to simulate neurons, while the Synapse one was based on SCDP. Both models were written on NestML and combined through the Python extension Nest with a generic code command. These are the main steps we followed to carry out our model, together with the main parameters and assumptions we work with. As mentioned before, changes in the postsynaptic calcium concentration drive plasticity mechanisms. The calcium concentration flow was modeled according to the differential equation shown here, and it was accounted for inside the neuron model. In our model, we assumed this concentration to be uniform all over the neuron, thus plasticity mechanisms is synapse location independent. Postsynaptic calcium enters dendritic spines through an MDR and BDCC, which are related to isolated pre- and postsynaptic spikes, respectively. Therefore, we decided to consider them as traces of synaptic activity.
Modeling the two currents, I and MDAR and IVDCC, was our main focus during the implementation of calcium concentration expression. The I and MDAR current equations were already accounted for in the Hilton-Oni model. This was the reason for our choice. This current and its related conductance change accordingly to the incoming neuron spike. Then, we modeled the IVDCC according to the Hutchin and Huxley formalism as suggested by Kindemi et al. Here, the calcium reversal potential was computed using the nurse potential equation. IVDCC equations were included in our neuron model as well. The C-star variable is used to capture frequency dependence of LTP. This variable allows calcium concentration to be integrated over longer time scales to drive plasticity. We reproduced this dynamic in our neuron model using the equation shown here. C-star directly influences the synaptic efficacy represented by the dynamic variable ρ, described by this formula. Values for the depression and potentiation rates were taken from the reference paper, as well as the two depression and potentiation threshold expressions. These expressions depend on C-star pre- and postsynaptic peak values obtained by running a separate simulation. Finally, the Heaviside function was modeled using flags. When a C-star value overcomes either the depression or the potentiation threshold, the function takes value 1, otherwise 0, allowing the raw function to be updated properly. Plasticity of neocortical synapses is associated on the postsynaptic side with AMPA receptor insertion and removal, which is dynamically linked to the raw variable. We substituted the predefined equation of G-AMPAR inside the Hilton-Oni model with the ones shown here. In our model, this conductance represents the weight of the synapse. It was updated at every synaptic event occurrence inside the neuron model and gave us an input to the synapse 1. The update consists of a multiplication for the vesicle release probability value, U, which we fixed at 0.5 following a deterministic approach.